It's been great so far to hear all of these talks, concentrated talks on neuroimmunology, especially on sort of these emerging roles of the peripheral immune cells infiltrating into the CNS. And today I'm going to talk a little bit about some of the data we have from our lab. Our lab, part of the work that we do uses the EAE, or animal models of multiple sclerosis, and some of the work that we're interested in protecting the brain, but also stumbled upon some interesting molecules that we think are involved in immune cell infiltration. As many of you know, multiple sclerosis is a devastating disease. It's a neuroinflammatory disease, and the cost of MS is astronomical, and the fact that it's an earlier onset disease, meaning that it really hits people in the prime of life. Many of them are college age and can't really maintain steady jobs. The cost of these drugs is pretty expensive. And disease-modifying therapies, although they've been wonderful, since 1999 was the first drug we had on the market for MS, interferon beta, and it has really improved the quality of life. Since then, we've have now about 13 drugs out on the market. It seems clear that we can reduce the number of relapses, but it's not clear that we can actually slow progression, although there are some drugs now that may be doing that. But ultimately, what we see in these patients with this disease is an ultimate disease progression and disability. Many end up in wheelchairs, and although the life expectancy isn't extended, certainly later in life their disabilities are quite pronounced. So obviously here you can see in this MRI just sort of an overview of MS. We do have these lesions. These are T2-weighted lesions, which are initially in the white matter regions of the brain. However, it's becoming more appreciated that there is either a secondary phase or as a consequence of axonal myelin degradation, a cortical atrophy or a neurodegenerative component of this disease, suggesting that we really need drug treatments that protect the brain. So what's known about multiple sclerosis is that the T cell is really thought to be the initiating event. That I shouldn't say that T cell, I should say T cell infiltration into the central nervous system parenchyma is thought to be a major initiating event. We can model this in animal models of MS. We do get this T cell infiltration. If we look at pathologically at postmortem tissue in MS patients, you do see T cells that are specifically primed for MOG and T1 reactive and T17 reactive T cells. And so the majority of drugs on the market either block T cell proliferation or activation in the periphery, and or also block their entry into the central nervous system. In particular, Tisabri is thought to block the blood-brain barrier to prevent these T cells from getting in. However, there are still relapses. So even on these drugs, eventually these cells, as well as others, macrophages, and there's a whole host that comes and ensues later on. Like I said, these are thought to be the initiating event, and then there's another inflammatory response that happens. But I'm not going to get into all of that, but I just want to point out the importance of having a drug that protects the central nervous system, meaning that we have drugs on the market that modulate these relapses or prevent immune cells from getting in. But then once those immune cells get in, we don't have anything selectively to protect the immune system. So that's really, protect the central nervous system. So protecting neurons from dying and protecting the myelin from degrading. So that's really where our lab became very interested in mechanisms of cell death in multiple sclerosis. In particular, our lab does a lot with glutamate in different cell types. And we were very interested in the role of glutamate and specifically if glutamate is a consequence of this inflammatory process. So in MS, glutamate is elevated in the cerebral spinal fluid in acute lesions and also in plasma of patients with MS. In the EAE animal model of multiple sclerosis, intraperitoneal injection of glutamate receptor agonists have been shown to attenuate the disease, as well as NMDA receptor antagonists, which has been shown by Howard Weiner at Harvard University. And also, if you take a global knockout of one of the major subunits that makes AMPA receptors active, or the glutamate receptor, AMPA, that there was a reduction in EAE severity, suggesting that there is an excitotoxic component in cytotoxicity, meaning that glutamate is selectively activating glutamate receptors in the brain. So, unfortunately, though, what we haven't done is looked at what the source of excitotoxic glutamate is. So we say, okay, there's evidence for glutamate and there's evidence for excitotoxicity in an autoimmune demyelination. But where's the source? And more importantly, why are we interested in the source is because those AMPA receptors and NMDA receptors and the glutamate receptors that mediate excitotoxicity don't really seem to be a feasible drug target because they're so imperative in normal cognitive function. Everything we do for normal cognitive functioning and movement, excitatory neurotransmission involves glutamate receptors. So having these as a logical target for any disease seems unlikely. So we became interested in this cystine glutamate antiporter system as a source of excitotoxic glutamate for a couple of different reasons. One, it's been shown to release glutamate in exchange for extracellular cystine. So that cystine comes into the cell and is involved in the biosynthesis of the antioxidant glutathione. But as a consequence of that exchange results in release of glutamate. And what's been shown in gliomas, which really are astrocytes sort of proliferated out of control. Astrocytomas or gliomas are brain tumors tumors in the brain, is that these cells, these astrocytes, will severely increase the cystine Xe transporter, and those gliomas almost become like an antioxidant powerhouse. You can't kill them because they have the glutathione. And so what happens is they release glutamate, which causes excitotoxic damage to the neuron through NMDA receptor-mediated excitotoxicity, which was shown by Harold Sontheimer at the University of Alabama at Birmingham. And if they blocked that process, so they blocked the death of the neurons, they could actually suppress the growth of the glioma, because the brain only has so much space. So if you don't kill the neurons, the glioma has no place to grow. And so showing the importance of this process here. And so this system Xe transporter is expressed in multiple cell types, including microglia and astrocytes. And so we all know that we get this reactive gliosis in the central nervous system, suggesting that part of this reactive gliosis may involve synthesizing that glutathione. So those reactive cells, those reactive glial cells don't die, which we don't really find much of those cells dying in the brain, usually neurons and oligodendrocytes. It's also upregulated as a result of oxidative stress, and oxidative stress is really an important mechanism in many different diseases, but specifically also autoimmune demyelination, suggesting that this process could upregulate system Xe on these cells. And it's been implicated in excitotoxicity in many other diseases, one being glioma, and we're also interested in now in MS. So as many of you know, we use the standard model for experimental autoimmune encephalomyelitis, and this is our scoring system for how we score these mice. What is interesting about this disease is you do take a MOG peptide or a peptide of myelin in conjunction with Freud's adjuvant, and this primes the peripheral immune system, in particular dendritic cells, to antigen-present to T cells, and causes a very specific MOG-reactive T cell phenotype, which then ends up into the central nervous system. In this model, the C57 Black 6 EAE model, you get a selective spinal cord disease, and in the SJL model, you get a little bit of disease, and we'll talk about that in a minute. You get an ascending paralysis, meaning that the tails become paralyzed first. It then moves up, and you get hind paralysis, first one limb, then both limbs, and then after that point, we do humanely euthanize the animal. So you have a consistent scoring system, and to understand how, in particular, any drug is being evaluated here. So the first thing we wanted to do is use our C57 Black 6 animal model of MS and test different inhibitors of the system XC transporter by doing intraperitoneal injection of these inhibitors. And as you can see on the left here, these are the animals that didn't receive our first inhibitor of system XC. And then when we introduced our first inhibitor, system XC, sulfasolazine, abbreviated by SAS, we do have a reduction in clinical scores, meaning these mice didn't present with the physical motor deficits. If we look at spinal cord sections using a gross stain of myelin, Luxol Fast Blue, in the spinal cord, white matter is on the outside, opposite of what it is in the brain. And we can see this blue staining here, loss of myelin without the drug treatment, and then in the box and in the bracket you can see a more enhanced blue stain, as well as blue stain here in the gray matter, which are also those axons that are a part of those neurons. And we can see a reduction here through our quantitative scoring. We used a second inhibitor of system XC. We did this really because sulfasolazine can also inhibit NF-kappa B, and as we know, NF-kappa B is a major regulator of inflammation, and we wanted to have a more specific inhibitor number one, didn't selectively target NF-kappa B at all. And again, we see the same result. And so we also went back and looked at reactive gliosis. We looked at staining with GFAP to show that we see in the spinal cord sections a reactive gliosis. We also labeled IBA1, which is, as Maya mentioned, IBA1 does also stain infiltrating macrophages, so we'll say that it's staining cells of the myeloid lineage. And again, we see a reduction here compared to controls with statistics. And also looking at system XC in general, using an antibody directed at system XC, we can see a reduction in the upregulation of system XC, specifically in reactive glial cells. So the first question that came about to us, especially since we collaborate with a lot of immunologists who are very interested in studying this model, is that we do intraperitoneal injection of these inhibitors. And so how do we know that we actually are selectively inhibiting something in the brain or selectively affecting a disease process in the brain versus attenuating the immune cells, the peripheral immune cells, from getting inside the brain? Because obviously, as we described, the T cell and initiating T cell response, if we block immune cells from getting into the CNS, we're obviously going to block inflammation. And so we went through a lot of very specific studies to count the number of immune cells getting into the CNS using facts and statistics. And interestingly, as a side note to this whole story, I will say, I'll get into the other data where we did look selectively in the brain, but it seems that system XC also has a different role besides blocking glutamate release and excitotoxicity, that it has a role in immune cell infiltration. And we also think that's glutamate related, and we don't have time to get into all of that now, but we'll just show you some of the data that we do have a reduction. So if we take out our spinal cords and do a total spinal cord count, looking for CD4 positive T cells as well as interferon gamma, which is a signature cytokine for Th1 cells, IL-17A, which is a signature cytokine for Th17 cells, as well as FOXP3, just to make sure that we didn't have some type of phenotype switch, meaning that we indeed have a reduction in the T cells, and it's not that our T cells are switching to a more T regulatory type, which is a non-pathogenic state. And we did that by looking at percentages of these cells, and we do not see a change in the percent of the state of the T cell, but rather a overall reduction in the number that enters into the spinal cord. We also wanted to make sure that this reduction in entry into the spinal cord wasn't just because the T cells for some reason didn't become as reactive as other T cells, meaning, for instance, that maybe the dendritic cells didn't present to them, or maybe they didn't proliferate in the spleens. And so what we did is we also took out the spleens from these mice acutely after the EAE at day 15 when you're at peak of disease, and we do not see a reduction in the number of T cells or their T cell pathogenic subtypes. We do not see a reduction in the T cell numbers proliferating in the spleens. And we also used Ki67 as a marker of proliferation, and we did not see changes in Ki67 positive cells. So this suggested that, again, this is somehow System XC is blocking immune cells from getting into the CNS. We also went a step further, and we did an EDU uptake, so we stimulated the cells with either CD3, MOG, two different concentrations of MOG, just to make sure our T cells were still reactive to the antigen, and they were. And again, we didn't see any changes here. So these data suggest a role for System XC in regulating immune cell infiltration into the CNS. And so we then became interested in, is System XC expressed on these infiltrating immune cells of peripheral origin? There's been a couple of studies to show System XC's on peripheral macrophages and T cells. And so we looked at these results in comparison to what we're seeing with IBA1 staining, and then used CD45 high-low and CD11B high-low populations to differentiate between peripheral macrophages infiltrating into the CNS versus resident microglia that are in the CNS, as well as T cells, or a lymphocyte population that is mainly T cells. And it looks like it's in all of those subpopulations. And this is a story then we want to follow up on selectively to know which subtype System XC is expressed on and how is it blocking this immune cell infiltration. And so just to confirm all of our pharmacological data, we took a mouse that has a mutant in the functional transporter, the System XC transporter, to see if if genetically deleting the System XC transporter confirmed our pharmacological data, because we all know that with pharmacology we have off-target effects. And again, here are our wild-type animals that have intact System XC. And these are animals where we've deleted the System XC. It's a global knockout, and we can see that these animals get the disease with the System XC transporter, and when we genetically delete the System XC transporter, these animals don't present till way late, and it's only a small percentage of the animals that actually even presented. So here, if we look at incidence, in these mice, they got a 60% incidence of EAE, and then when we genetically delete System XC, you only have 20%, but that is severely delayed, suggesting that we definitely restrained this EAE disease in these animals that lack functional System XC. So now we want to get back to this idea of excitotoxicity and what's going on in the brain. So we wanted to further explore the role of the System XC in CNS protection. And so we thought the best way to do that is let's let the immune cells get into the brain, let's have the normal disease inside the brain, and then let's introduce the inhibitors of System XC and see if we can protect the brain. And so we used a model of relapsing remitting EAE. This model is thought to better represent the human condition for two reasons. One, it gets a relapsing remitting form, which means it gets the disease and then it gets better, it gets the disease and gets better, and that's what most patients with MS are diagnosed as, relapsing remitting. It also gets disease or brain pathology. So the C57 Black 6 animal model, pathology is, for the most part, predominant to the spinal cord, and then the SJL model, you get spinal cord and brain pathology. And so, what we did here is we induced our EAE in our this is an different background of mice, SJL mice. We allowed them to have their relapse, and while they're having their relapse, we introduced a System XC inhibitor. And again, we see a reduction in disease. And we also see a reduction in myelin loss. This is without PBSs or saline injections were our vehicle. So this is our control animals here, and these are SAS-treated animals. We then looked in the spinal cord, and again, we saw a decrease in reactive gliosis, but in these animals, as I mentioned, they get brain pathology. So we did using the area fraction technique and taking pictures and looking at the area staining of these reactive cells versus not, so we can quantitate this. Again, we have a reduction in IBA1 and GFAP, and also looking at Luxol fast blue stain. If we look at the corpus callosum, and in areas of the corpus callosum right above where you have axonal projections into layer 6, we see less myelin damage. Then we wanted to understand this mechanism and actually see if these T cells are somehow involved in producing glutamate that can cause this excitotoxic damage. And the best way to look at this mechanism would be to do, obviously, co-cultures and understand how this system XC transporter is releasing glutamate, and if indeed can we reproduce this oligodendrocyte death that we're observing. And so what we did is we specifically isolated MOG-reactive Th1 and Th17 cells from the spleens of mice that were induced with EAE, and then we co-cultured them with our mature oligodendrocytes. So we culture oligodendrocytes and add different growth factors to make them mature myelin-producing oligodendrocytes in the presence of microglia, or in the presence or not in the presence of microglia. And what we found is that specifically Th1 cells did not kill mature oligodendrocytes unless microglia were present. And that was the opposite for Th17 cells. For Th17 cells, we saw direct death of oligodendrocytes with or without microglia. Then we were interested in nitric oxide release, since we know that microglia play an important one of their modes of toxicity is through nitric oxide. And again, oxidative stress, producing an oxidative stress environment is what upregulates the system XC transporter and gets it releasing glutamate. And what we found here is that Th1 cells definitely potentiate nitric oxide release from microglia, and Th1 cells selectively signal interferon gamma and TNF alpha as their signature cytokines. And so we wanted to see if this was a cytokine-mediated effect. And so we repeated this experiment in the presence of antibodies against TNF alpha and interferon gamma, and we could selectively reduce the amount of nitric oxide produced. We then went on to look at cell death. If we take our interferon, if we, again, remove the T cells, but now take the cytokines that they signal and culture that with our microglia and our oligodendrocytes, can we reproduce this cell death phenomenon? And we can. So here is our cell cultures, and we use olig-2 to stain for oligodendrocytes because it is hard to see these cells in the process of dying, and we count the number of cells left over after a 24-hour period. And we see that we get the most cell death with interferon gamma TNF alpha stimulated microglia, and that is the only cytokine of all the cytokines that the Th1 and Th17 cells signal that cause this release of nitric oxide. So we then set up this same paradigm and wanted to see if we could reproduce this classic excitotoxic phenomenon. And we co-cultured our cells again with interferon gamma TNF alpha stimulated microglia. And we looked at cell death, and we definitely get cell death, and it's blocked by an inhibitor of the glutamate receptor AMPA. So this indicates that this is an excitotoxic phenomenon, since we can protect cell death by a glutamate receptor antagonist. And you can see here in this paradigm that we do have an increase in the system XC transporter as well in the microglia, and which is we quantified by densitometry. And we also looked at the release of glutamate in the supernatant. And what we found is that when, again, system XC transporter is upregulated, we have a huge increase in extracellular glutamate that is inhibited when we block with system XC, showing that indeed in this paradigm, system XC is releasing glutamate. So then we went a step further and used our same cell culture paradigm and saw that we can actually protect these cells from cell death with two different system XC inhibitors. So those data suggest that indeed the system XC transporter is involved in excitotoxic mechanisms, and we can block the source of glutamate in the CNS, and that it also plays a role in immune cell infiltration. And so one last experiment that we did to sort of complete the story, and we're still working on, is that if the system XC transporter is really involved in releasing excitotoxic glutamate, and the other studies, one did intraperitoneal injections of the glutamate receptor antagonist. It's not clear whether or not they may have been attenuating some immune cell response. And the other story where they did a global knockout of an AMPA receptor. AMPA receptors are on neurons and many other cell types. So what we wanted to do is selectively delete AMP receptors on the myelin itself. So AMP receptors are not deleted anywhere else, but only on the myelin, and see if we again produce this EAE model that we can actually see protection of the brain and the myelin. And so what we can see here, in mice that do not have the AMP receptors deleted in myelin, we have myelin degradation. This is DAB staining with myelin basic protein. And we can see loss of myelin here. We didn't look at axon damage here or not, so that's something else we're going to look at. And then here in our animals where we selectively delete the AMP receptor from the oligodendrocytes, we have protection of this myelin here. And we haven't done FACS analysis yet, but our preliminary ICC says we haven't changed the T cell infiltration at all. And so we're going to look further into that, but for the most part we are seeing that deleting these AMPA receptors protected the myelin, suggesting that there's an important excitotoxic component in MS, and that we are now going to look at that further as part of this neurodegenerative component that seems to be an emerging topic of study in MS as well. So inhibiting system XC attenuates CNS infiltrating immune cells during EAE, but does not reduce proliferation or activation in the spleens. And inhibiting system XC ameliorates the EAE clinical symptoms and reduces demyelination and reactive gliosis after immune cell infiltration. And the role of system XC on these different immune cells is unknown, but a future exploration for our lab. We'll also like to acknowledge the people in my lab, mainly Kirsten Ivanuk and Brandy Baker, who did the work, a graduate and a postdoc student, as well as many wonderful collaborators, in particular the Weaver Lab, and also the Raman Lab, two immunologists we collaborate with at UAB, and Sandra Hewitt at University of Syracuse, who provided us the System XC knockout mice, and of course funding sources.